Hello, everyone. It's pretty hard to pass after Nathaniel from Chicago, but I will tell my kids you're Michael Jordan. So I'm Sébastien from Stéphania Milvoy's team here at CRCT. So it's the base team of CRCT, of course, because we work with accelerators. And this work, it's QUADRatlas, so it's a database about RNA structures and RNA-binding proteins that we made in collaboration with Eric Dassi's lab at CBO in Italy. This work is about structures called RNA G-Quadruplexes. So these RNA structures are coming from G-rich sequences, like you can see here, and in some cases they can form these quadruplex structures. This is not working. So thanks to hydrogen bonds between the guanines, and so these structures are in equilibrium with the G-rich sequence, and it can be regulated by some transactive factors like ions. For example, K+ can stabilize these structures, or ligands. So we have these two ligands, for example, that stabilize the structure, and it can also mostly be regulated by binding proteins. So you have two types of proteins. So RNA G-Quadruplex binding proteins that prefers the folded state of the structures, and G-rich binding proteins that binds the sequence when it's unfolded. So you can find the structures in every type or every classes of RNAs, and the enrichment of the structure is basically in both of the translated regions. And these structures can be found in the nucleus or cytoplasm. So the structures have been described as a regulator of every step of post-transcription, which goes from the transcription of DNA to free mRNA to its translation into proteins or its degradation. So this work reported a lot of interaction with RNA-binding proteins that controls the folding state of the structures. And for example, during translation, if the RNA G-Quadruplex is folded, it can block the ribosome and block the translation. And some RBP, like ABKs, can unfold the RNA G-Quadruplex and allows the translation of the mRNA. So this has been reported for a lot of cellular processes, and including a lot of pathologies too, in cancer, for example. So that's why it's really important to explore all the interaction between RBP and RG4s. So the gaps for now is to explore all this interaction. So the two main questions are which RBP binds a defined RG4, and on the contrary, doesn't RBP bind multiple RG4 containing mRNAs? And of course, the link of this interaction with cellular processes and disease. So these are what we can have for databases for now. So we have prediction tools of RG4, which predict RG4 according to the sequence you give. We also have databases about the ligands of RG4. We can fold or unfold the structure. Some databases focuses on the structure of the RG4. And of course, we have databases of the non-G4 in the transcriptome. So here the limit is that none of these databases deal with the interaction with RNA-binding proteins, and we didn't find any integration of prediction and experimentally determined RG4. So with QUADRatlas, we find a way to link RNA binding proteins and the target RG4s. So I will explain the data of the database and then every function that we made. So first, of course, we took RNA G-Quadruplexes sequencing. So I will explain how we can get experimental sequencing of RG4. So basically, in two different conditions, we have the light chain ions that unfold these structures and the K plus ions that stabilize the structure. So when we do the reverse transcription of the RNA, the ribosome is the transcriptase is blocked in the structure that gives a short cDNAs, and then with the sequencing, you can find high coverage in 3' of the structure. So that's basically how you can sequence RG4s in the transcriptome. So we have like 40,000 RG4 sequence in the database, and as I told you, it's mainly enriched in the untranslated regions. And then we also added predicted RNA G-Quadruplexes. So we used 3 different tools that takes into account the number of G different, the bunches, the mismatch, and the loop length. So here is an example of the calculation of a score to predict an RG4. So basically, it's the number of iterates minus the score for the mismatch and minus a score for the bunches. And after this, you have calculation for the loop length. So we use three different tools with their own calculation process and thresholds. Again, we find 50 to 75% in the UTRs. And we also calculate a consensus of these 3 tools to have the most relevant RNA G-Quadruplexes. Okay, so now we have the data for the RG4s, and I will explain you the data for the RNA-binding proteins. So there is 2 types of data. The first one is to determine the binding proteins that specifically binds to these structures. So this is obtained by RNA pool dance or specific studies. So RNA pool dance is basically an RNA with biotin, which contains an RG4. And when you pass a cellular extract on it, you can isolate and purify binding proteins that fixes these structures and then identify them by mass spectrometry. So in this database, we use 8 of these RNA patents and 95 publications of specific interactions. So then we also included binding sites of 150 binding proteins. So the binding sites are determined by cross-linking and immunoprecipitation. I will not explain in details, but this will allow us to compare binding sites of RNA binding proteins, not specifically the ones that binds RG4, but we can compare these binding sites with the RG4 sites in order to have some hints of new interactions. So in this database, we set an exploration with JBox2 of all this data, so you can look at every interaction. We also put some analysis functions that I will explain, and of course we can download everything. And yeah, I will start to explain this. So this is how it looks. It was developed with Django for the backend and Angular material for the front end. So the first two functions. First, yes, we put some functions for RG4s and some functions for the RBPs before putting everything together. So these first two functions have to look in a transcript or a list of transcripts if you have RNA G-Quadruplexes structure in it. So this is an example. So you choose the data to explore, whether it's predicted or experimentally determined data. And then we give you every details and also the sequence that you cannot see here of the RG4s that have been found. And then for the enrichment function, you give a list, and we perform a Fisher's exact test to see whether this list is enriched in RNA G-Quadruplexes. And we also added this table to this function. So we also give you the binding site of RG4 binding proteins that has been found in your transcript. So you can have a list of the interaction with binding proteins. So we have the kind of the same function for the binding proteins. So it's search RG4BP, where you can find information about your RG4BP. Yeah, this function finds the targets that we found in the literature of your RG4BP, and then we take this targets list and we give you the potential RNA G-Quadruplex that we found in these targets. And we have also an RG4BP enrichment function that finds the RG4BP that potentially binds to a list of targets and gives you again a p-value based on a t-test, a Fisher test. Sorry. So this is like the main function of the database. It's the overlap one. So its main goal is to find a new interaction between RNA G-Quadruplexes and binding proteins. So not coming from the literature. So here you can enter an RNA binding proteins, and this tool calculates the interactions, the intersection between RBP binding sites and RG4. So we give you at the end a table with every binding site of the protein and every RG4 that overlaps these sites. We also put a prediction tool. So with the three tools that we use to predict every RG4 in the human transcriptome. So you can choose every threshold of the three tools. And the idea is to compare the three tools for a given sequence. So you can see clearly in the browser or in the table. And then you have a big browser to look every tracks that we have that can be useful to check for a specific gene. So you can look at it and look at every RNA G-Quadruplexes and every binding site of the protein that we have. Yeah. And the thing that I use the most actually is a batch download. So you just put a list of genes and it gives you every annotation about the gene, the disease associated to the genes or cellular processes. And if the list contains the protein, it gives you the RG4s that can be bound by the proteins. Also, of course, the RG4s contained in the input genes. or transcript, and the binding sites of the RBPs that are in this transcript. And so it's really useful if you need to compute after this and do analysis on your own. So that's it. So for the evolution of the database, we want to add other organisms, because for now we only have human. So we do this with some collaboration if we have some. And we want to add the newly discovered G4-binding proteins to the dataset that we plan to do every year. And we want to compute more statistics and actually on the overlap functions. So that's it. I want to thank everyone in the team, especially Sébastien and Leila, Pauline, and from Team Italy, so Eric, Eliana, and Andrea who contributed. And thank you.